Rajesh sir,
Yes,
so we can start now.
yeah. Good evening, all. I heartily welcome to all of you in this faculty development program based on statistical analysis in academics and research. Now, I welcome our today's guest speaker, Honorable Dr. Ojoyo Samsidan, who is working as Associate Professor in the Department of Geography, Federal University of Technology, Minna, Nigeria. Now I request our honorable chief guest to start his presentation and talk.
Thank you very much. Good evening, everybody. Though it is afternoon in Nigeria here, I hope you are hearing me.
Yeah, we are getting you, sir. Please go ahead.
Okay. Thank you very much. When we talk about statistical analysis for research, it depends on the types of research. And when statistics is involved in any form of research, the researcher needs to take into consideration the objectives and the research questions that need to be analyzed. The objectives of the research, the problem statement will guide the researcher on the form of statistical analysis to utilize. When data are collected, data that will be collected will be based solely on the objectives of the research work. Most especially in quantitative research, it is very important that the researcher develop an instrument to gather his data. Some of these instruments may be presented in form of in a questionnaire. This questionnaire will be presented based on the objectives of the research. And as soon as this questionnaire is presented, the researcher will have at the back of his mind that the questionnaire are going to be addressed are not going to be an open-ended question. An open-ended question gives a lot of variant opinions of observers. So the analysis of this may be a little bit cumbersome. So what the researcher will do is to streamline its questions so that the questions that will be involved are those that will be close-ended. So it is important also that the researcher takes into consideration the various objectives. The research questions are going to be taken into consideration so that when the data are now obtained from the field, various statistical methods can now be used to analyze the data. Some of the statistical methods that could be used may be just descriptive statistics, those ones that will show the biography, the biodata of the respondent. Then in answering the research questions and the hypotheses, if there are any, inferential statistics could be used. Both inferential and descriptive statistics will go a long way to portray the major objectives of the research. And when these are done, at the end of the day, to anybody that is picking up the research work, we know vividly where the research is heading to, what are the achievements, what are the goals that have been achieved. So when this is done, the researcher will be so much interested in the fact that he has actually use a research method. Thank you.
Yeah, okay. Thank you very much, sir.
You're welcome.
We are very thankful to you for your valuable time to us. Now we have our next speaker, Mr. Mayur Patil, and I request Meenal Masti ma'am to introduce our today's speaker, Mayur Patil.
Thank you, sir. Good evening. It is always said, what we learn with pleasure, we never forget. And such is this FDP arranged by International Journal of Microbial Sciences. And I am here to introduce our today's first speaker, Mayur Patil, sir. Sir is pursuing PhD in botany and has completed his master's in botany with CSIR NET, ICAR NET, SET GATE, and currently working as assistant professor, Department of Botany, SNJB's KKHA Arts, SMGL Commerce, and SPHJ Science College, Neminagar, Chanwad District, Nashik, Maharashtra. Mayur sir is working on a project titled Nutritional Attributes of Some Wild Legumes. He has published about 4 abstracts and 2 research articles in journals. Sir has attended 1 week online FDP on mathematics for biological sciences organized by HC Savitribai Phule, Pune University. Some of his MOOC courses completed by him include Biostatistics and Mathematical Biology Swayam, offered by Central University of Punjab, Enhancing Soft Skills and Personality Swayam, offered by IIT Kanpur, and many more. Patil sir has been honored with different awards and achievements like selected to MPSC, Assistant Professor in Botany, Maharashtra Education Services MES, Group A 2015, topper in MSc All Streams, J.K. Patil Prize recipient. While the courses taught by Patil sir include computational botany, plant biotechnology, plant morphology, and anatomy, industrial botany, horticulture, and floriculture, principles of plant science. Now I request Mayur Patil sir to take over the session. All over to you, sir. Thank you.
Thank you, Minal ma'am, for such a nice introduction. First of all, I would like to thank Dhakne sir for giving me this opportunity to give a talk on such a nice program, FDP, that is Statistical Analysis in Academics and Research. Okay, so I'm very much thankful to all the organizer team for such a nice opportunity. And Meenal ma'am has introduced me in a very nice fashion, and thanks again for that also. So without wasting the time, we will start our presentation. Sir, please allow me to share the screen, please. Dakne, sir.
Yes, sir. Wait.
Sir, please allow me.
Yeah, we allowed, sir.
Yes, yes.
Please go ahead.
Okay, sir. Now, is it visible, sir?
Yeah, yes.
Okay. So, with warm heart, I would like to welcome all the dignitaries of a group, all the speakers, all the anchors, and not last but not the least, all dear participants to this 1 week faculty development program on statistical analysis in academics and research organized by International Journal of Microbial Sciences, IJMS, in collaboration with Sushila, Shankarrao, Gadhave, Mahavidyalaya, Khandala, Dist-Satara. So today I have been assigned a topic to talk on mean plus standard deviation, median, interquartile range, and bar charts. Okay, I'm going to talk on this topic, which will be very fascinating and wonderful experience. What I have achieved through these years, I would like to share my knowledge with all of you people so that it will be beneficial to you in the academic or research purpose. So it is a brief achievement that I have achieved up till now, and many more to come. It's nice privilege to be a part of this FDP. Thank you very much for such a nice and giving me this opportunity. Okay, so without wasting the time, we'll start the presentation. So basic thing is why we should have the knowledge of statistics in research. So basically, if we see why we should have knowledge, so first thing which comes in our mind, that research article, when we read the research article, there must be some part which is related to the statistics, and which is even we are from biology background, even though the statistics is applied in all the streams, in physical sciences, in chemical sciences, in life sciences, botany, zoology, but the people from biology background find it somewhat difficult to understand the statistical part of that particular research article. So basic thing to learn the statistics is to understand the statistical portion of most of the scientific article. That scientific articles, no research article will get complete in true sense if proper statistical method is not applied in that research article. So to understand the research paper wholly, it is very important to know the basic statistical methods which have to be applied in that research article. Then second thing, to plan and design your own research project and experiment. So to properly plan, to design in your sense, that RBD is there, randomized block design. There are many different methods by which you can design the experiment. So how to design the experiment, for that the knowledge of statistics is of utmost importance. Then last point is to know which type of statistical test has to be applied to a particular type of data sets, means which type of data you are having, and to that data which type of statistical test is applied, we must know, and for that the knowledge of statistics is essential. Then you are seeing this man with a lot of question mark in his head, that is confused about how to apply the statistics in research article. What are the different ways? Because basically, if we consider it as a man from a biology background, he don't have that much knowledge of statistics, but as a part, when we have to publish the article, then some statistical methods needs to be applied. So he's thinking that how to apply the statistics in the research article. So the question may arise in his mind, that should I learn whole the statistics, which is slightly different from our biology background, or should I seek for the expert? But according to me, according to my opinion, whatever you do, whatever research you do, you are the best person to represent that research in front of the peoples, because no one knows your research better than you. So when you are going to represent it, when you are going to write a research article, each and every part of research article should be properly known to you. Means you don't require any sort of experts, according to my opinion, because the people tend to run away from the statistical things, that they seek for the experts. From the people from the statistical background, they take the help. It is not it is good to take the help, but it is also better to learn by yourself only. So I think it is better to learn the statistics, because if we know each and every part of a research article, the statistics is also part of that research article, and we must know it. Okay, so let us move forward. That I have taken some reference here. Let me take highlighter. See, this is a result table which I have taken from a research article which is published in 2009. I have given here the references. B. Bhagia and et al. His co-workers had published this article in 2009. But if we see this research table, you will find something like 53.77. Okay, this is given in that result. So what actually it is representing? So it is given at the base. Okay, if you see here, values are expressed as mean plus or minus standard deviation. SD means what? SD means standard deviation. And what is N there? N is the number of set of experiments that is being performed, or that had been performed in that calculation of that sodium. For example, sodium. So what is that? Mean 53.77 is your mean, plus or minus 3.33 is your standard deviation for the set of observation. Means what? You have performed that same experiment 5 times. You have taken 5 readings. You have calculated 5 different values, and you have taken the mean of that 5 different values, and you have calculated the standard deviation of that 5 different values or 5 different observations. So how this is calculated, that is of our interest. That is what we are going to see in our as the lecture progresses. It's very interesting and fascinating. When you will complete this lecture, you will, I am sure that you will definitely understand how to calculate that. So let us move forward. What we have to study? We have to study the standard mean. How to calculate the mean is very simple, but how to calculate the standard deviation for n number of observations? Let us move forward. So before knowing that, we must know some basic things regarding the statistics. What is statistics exactly? Statistics is a method of interpreting, analyzing, and summarizing the data. Means it is divided into, when we do something survey like kind of things, what we do, we go and collect the data. Okay, after collection of the data, we properly classify the data, we properly arrange the data in some sort, and then we properly represent the data. There are different methods of representation of data sets. For example, you can represent in tabular form, you can represent in graphic form like histogram, frequency polygram, cumulative frequency curve, or like a scatter diagram or dot diagram. In graphic form also you are able to that represent that data. And diagrammatic type is also there, in which Venn diagram, bar diagram, etc. you can use to represent that data. So basic thing, you do what? You just collect the data, you just organize the data, you just represent the data in a proper way, proper fashion, and then you interpret it, you analyze it, and based on that analysis, based on that interpretation, you give some meaningful conclusion from that data. So this is what statistics is all about. Statistics is very important, because without applying statistics, we cannot properly state something significant. Okay, to state something significant, we must know how to apply the statistics. So basically, what is statistics that we have discussed? Then after that, we'll study what is biostatistics. Okay, what is biostatistics? Biostatistics is nothing but, it is the applied branch of the statistics. It is the applied branch of the statistics which which apply the statistical methods which apply the statistical method in biological sciences. Means what? Biostatistics is an applied branch. It is the applied branch. What biostatistics does? Biostatistics apply whatever the statistical methods that are present in the statistics. We apply the statistical methods appropriately in a biological sciences. Okay, so there are basically two different parts in which statistics is divided. First one is your descriptive statistics, another one is your inferential statistics. Okay, statistics can be classified, can be described in these two different forms. First one is your descriptive statistics, another one is your inferential statistics. What is descriptive statistics? In descriptive statistics, measure of central tendency, measure of dispersion, it comes. Means what it does, it measures the it calculates the measure of central tendency like mean, mode, median, as well as it also measures the measure of dispersion, which includes mean deviation, quartile deviation, standard deviation, variance, and range. So all this comes under the descriptive statistics, which describes that data. What is inferential statistics? Whatever the calculations that are being made, like calculation of mean, mode, median, after calculation, what we do, we compare the different means of different populations or different samples. So comparing the mean comes under the inferential statistics. Infer means we infer something from that, because we compare means and we draw a meaningful conclusion from that. So that comes under the inferential statistics. For example, ANOVA, then t-test. If we want to compare a mean of more than two different samples, then we use the ANOVA method. And if there are two different samples only, then we use the t-test, student t-test. So that is about the parts of the statistics. So briefly, we will discuss what are different measures of central tendency. So there are different measures of central tendency. What is central tendency? Basically, we must know what is central tendency. Central tendency, or a measure of central tendency, is a central or representative value of particular data set. Means you have given 5 different sets of observations. Say for example 2, 5, 9, 4 and 3. 2, 5, 4, 9 and 3. And these are 5 different sets of observations, and you are asked to measure a central tendency or to state a central tendency. So what will we do? We will find out one value which will represent that whole data set. So that one value, that representative value, that central value becomes your mean. That becomes your measure of central tendency. So there are 3 different types of measure of central tendency. First one is your mean. Mean, I think you have learned it in your school days also, how to calculate the mean. Mean is simply the average. Average means what? How average is calculated? Average is calculated like sum of all the values, and it is divided by the number of values. In this fashion, mean is being calculated. Then second measure of central tendency is your median. What is median? Median is simply, it is the middle value. Middle value means central value, not exactly central value is correct. Middle value means there are, say for example, 9 number of observation. So the 5th one is your median, but there is a condition that you should arrange that data in the ascending order, and after that the middle value becomes your median. And last one is your mode. What is mode? Mode is very simple. It is the most repeated or highly repeated value. Okay. So actually median and mode, they represent the positional averages only. But mean takes into account every single observation. That is the difference. So these are some of the measures of central tendencies: mean, mode, and median, which is a part of descriptive statistics. Then second part, which is most important from today's perspective, that is the measure of dispersion. See, Measure of dispersion. What is measure of dispersion? Let me tell you first. If we define measure of dispersion, it can be defined as a deviation or a scatter or a spread of individual observation from its central value. Means whatever your central value means, what central value means your measure of central tendency, but how the single individual observation deviates, spread or scatter from its central value, that is your measure of dispersion. Okay, there are four different types of measure of dispersion. Quartile deviation is also there, but these four, like mean deviation, second is your standard deviation, third one is your variance, fourth one is your range. Now let me take one example by which you can understand the importance of measure of dispersion over the measure of central tendency. If I have given here three different types of data sets, 60, 60, 60, 60, 60. There are five number of observations are there. Second data set is also having five different number of observations. Third data set, it is also having five different number of observations. But if I ask you to calculate, if I ask you to calculate the mean of these three different data sets, how will you going to calculate? Just you will add up all these number and divided it by the number of observations, that is in this case it is 5. Okay, and if you calculate for first, it comes as 60 only. It comes as 60. Mean comes as 60. You can represent mean by a symbol X bar. You can represent by this. I am writing with the help of mouse, therefore there may be some flaws in the handwriting because it is a mouse. Okay, but I will try to write in a proper manner. If first thing, first data, number of observation, if we see, and if we calculate the first mean for first number of observation, it comes as 60. If we calculate X bar for second number of data set, 30 plus 50, 80, 80 plus 85, 165, 165 plus 75, 240, and 240 plus 60, it becomes again 300. 300 divided by 5 is, it again comes the 60. It again comes 60. Okay. In third case, if we see third case, what will be the mean in third case? In third case also the mean comes 60. How? 10, 60 plus 10, 70 plus 90, 160, 160 plus 90, 250, 250 plus 50, 300, 300 divided by 5, it becomes 60. So what my point to say is, even if these three different data sets are having different number of observations, in first case all are 60, but in second case it is different from the first, in third case it is again different from first and second. So even if there are different number of observations, even if such case is there, their mean, their central value comes the same. So we cannot predict from the central value that data sets are same. Okay, so and this flaw is limited or it is eliminated in case of measure of dispersion. What happens in measure of dispersion? As we will come to previous slide. So what is measure of dispersion? It is a deviation or a scatter or a spread of individual observation from its central value. Means what? Here we have to calculate the central value first. After that, we have to take into account each and every observation. We have to find the difference between every observation and the mean. For example, in first case mean is your 60, but the difference between every observation and mean is 0. In first case, 60 minus 60 is 0. So that is 0. So secondly, but there is a difference. 60 minus 30 means 30. So 30 is at a distance of 30 from 60. Listen carefully what I am telling you. 60 is your central value, is your mean. How far 30 is from the mean position? It is at a distance of 30. How far 50 from the mean position? It is at a distance of 10. How far 85 from 60? It is at a distance of around 25, not around exactly 25. Okay. So here in case of measure of dispersion takes into account every observation. So it is very significant thing. So that is about the measure of dispersion, and the most famous measure of dispersion is your standard deviation. So what is standard deviation? Standard deviation is nothing but it is defined as a degree to which each and every observation deviates from its mean value or its central value. Okay, or it can also be defined as a square root of variance. Okay, that is another definition. But what is main definition? Degree to which each and every observation deviates, scatter, spread from its mean position. That is what standard deviation is. Standard deviation means, see, I am linking this concept with what we have seen in the, I think, 4th to 5th slide. That is, I have given a table, a research table from a research article where the values were expressed as mean plus or minus standard deviation. So I am relating this with that only. Okay. So first thing we have discussed about mean. Secondly, we are discussing now about standard deviation. How the standard deviation is calculated, then that is of utmost importance. So standard deviation is calculated by means of this formula S. S is equal to square root of summation x minus x bar bracket square upon n. In this way, we can calculate the standard deviation. If the sample size is less than 30, then you can use n minus 1 as it is given in different reference books. That if the sample size is less than 30, then you can use n minus 1. So this is the formula. S is equal to summation of x. X is the variable. Okay, variable or observation. What is x bar? X bar is your mean. X bar is your mean. Say for example, you are having 5 different type of observations. You will going to calculate the mean. That will be your x bar. X minus x bar bracket square and its summation, and which is again in the square root divided by n. This is the formula by which we can calculate the standard deviation. Now let us consider the example that say for example, you have performed one biochemical experiment. You have taken the absorbance, you have taken the reading, then according to that absorbance you have calculated some values, that results, and say that values are your these values 5, 7, 2, 6 and 10. There are 5 different values, 5 different sets of same experiment you perform, because you cannot perform only a single experiment and state anything from that, because error may happen in that case. So for that, either you should perform it in triplicate, either you perform in 5 times. Okay, so in that case, in that research paper, it was performed in 5 times, therefore n was equal to 5. Okay, so consider the same situation where you took the absorbance of that, you calculated the value, and that values are these values 5, 7, 2, 6 and 10. Now what we have to do, we have to represent that values in the form of this mean plus standard deviation. In this form we are going to represent it. So how will we going to represent? So basically, first thing, while calculating the standard, you have to calculate 2 things. First thing, you have to calculate the mean. Second thing, you have to calculate the standard deviation. So first thing, we're going to calculate the mean. If we consider here how the mean is calculated, mean is calculated by this formula. X bar is equal to summation of x divided by n. So this is the formula for calculation of mean. So if we observe this data 5, 7, 2, 6 and 10. If we calculate the mean for that, 5 plus 7, 12 plus 2, 14 plus 6, 20 plus 10, 30. 30 divided by 5, it comes your 6. It comes 6. So x bar in every case, it comes 6. Okay, it comes 6. So it comes 6. So in this way, x bar is calculated. See, we have to memorize the formula of standard deviation. How the standard deviation is calculated? S is equal to square root of summation of x minus x bar bracket square divided by n, or you can take n minus 1 also. Okay, if the sample size is less than 30, in this case it is less than 30, so we'll take n minus 1. Okay, so in this case we have to go according to the formula. Formula requires x bar, so we have calculated x bar. Formula requires x minus x bar, so we'll calculate x minus x bar. X minus x bar, 5 minus 6, it becomes minus 1. 7 minus 6, it becomes 1. 2 minus 6, it becomes minus 4. 6 minus 6, it becomes 0. 10 minus 6, it becomes 4. Okay. Then next thing in formula is the bracket square. So we're going to square it up. Minus 1 into minus 1, 1. 1 into 1, 1. 4 minus 4 into minus 4 plus 16. Plus 16. Then 0, it becomes 0. And last but not the least, 4 into 4, it becomes 60. So in this way, the x minus x bar bracket square is calculated. Now, what is given in that formula? In that formula, it is given as a summation. So here we have to take the sum. It comes your 16 plus 16, 32 plus 1 is 34. So your numerator value comes as a 34. Your numerator value comes as a 34. So this is the formula unto which we have to apply the formula. So S is equal to square root of x minus x bar bracket square, it comes your 34 divided by, in this case, n minus 1. We have to take n minus 1. So here in this case, how what is your n? N is your 5. 5 minus 1 means 4. So again in this case, here we have to add n minus 1 if the sample size is less than 30. So here 34 divided by 4, and which is under the square root, it comes your 8.5. 8.5. It comes your 8.5. Now if you take the square root of 8, then it comes your 2.91. I have calculated by using calculator. It comes your 2.91. See, now how the representation we wanted? We wanted representation as mean plus or minus standard deviation. Mean is your 6 and standard deviation is your 2.91. So how that data is going to be represented? The data is going to be represented like 6 plus or minus 2.91. So in this fashion, we're going to represent that data in the form of mean plus or minus standard deviation. That in this case, 2.91 is your standard deviation. See what it exactly means. It exactly means your mean is 6, but your individual observation tends to vary up to 2.91, either from right side or either to the left side. Means it can be 8.91, 6 plus 2.91, or it can be 6 minus 2.91. So in this way, that mean and standard deviation is expressed. Now, dear friends, I have explained you the basic concept, how this from raw data or from the observation, how we're going to calculate the standard deviation, the mean, and how to represent it. But it is a manual method of representation. But nowadays, there are many softwares which are available in the market, like your SPSS, Statistical Package for Social Sciences, or there is a Minitab, there is a DraftPad Prism, okay, Statistica is another software. So these softwares are there which will give you the ready results. You don't have to calculate, but for understanding purpose, how the standard deviation is represented, this theoretical knowledge is must for all of us. Okay, so that is about mean plus or minus standard deviation, which I have told you in the previous slide, that research article, that table. So in this fashion, that table is calculated, or that values are calculated. Now, there is another concept of standard error of mean. You may find in some research articles that instead of standard deviation, SEM, standard error of mean, or only simply standard error, SE, it is also represented, means the data is also represented in the form of mean plus or minus standard error or standard error of mean. But my dear friends, when you want to show a variability between the samples, between one single samples that you have taken, one set of observation, if you want to show a variability between that only, you should use, or you must use, the standard deviation and not the standard error of mean. What is standard error of mean? What it actually represents? It represents the precision, the accuracy of a sample mean to that of the population means. Means there are different number of sample. One sample is from, say for example, Nasik district, one from Mumbai, one from Chennai, like that, and you will going to calculate the mean of each, and you will compare the mean, and you will take a mean of mean. Okay, that will be your population mean, and you will compare each and every mean with that population mean. That will become your standard error of mean. But fortunately, if you want to calculate SEM in single samples also, it can also be calculated like standard error of mean. Simply what you have to do, you have to calculate the standard deviation, and after that you have to divide it with the help of square root of n. So if we calculate it, we will get standard error of mean. I hope you have all have understood this. Then next topic is interquartile range. But before knowing the interquartile range, we must know what is range actually. Range is also a measure of dispersion, as we discussed earlier, as we have seen in earlier slides. So what is range? Range is the difference between maximum and minimum number of observation. It is a very simple measure of dispersion. Very simple. For example, if I give you one example like 1, like 3, like 5, like 15, like 20. Okay. So there are how many number of observations? There are around 1, 2, 3, 4, and 5 number of observations. Then if you are asked to calculate the range for such data set, how will you going to calculate it? Simply you have to find out, you have to trace which is the maximum observation or highest value of that observation. In this case, it is your 20, or the largest value, you can say L. L stands for largest value. L is your largest value, which is 20. What is your smallest value? 1 is your smallest value, it becomes 1. So how the range is calculated? Range is calculated by the formula L minus 1, which is 20 minus 1. In 20 minus 1 means what? It comes your 19. So 19 will be your range for that particular data set or set of observations. But my dear friends, if we carefully observe that, we see that range takes into account only two observations, not all the observations. 3, 5 and 15, they are not taken into account. So there is some inadequacy, there is some flaws in that calculation. So we can say drawback, it also gives something, but it is not proper, it is not exact. Okay, it only takes into account two extreme observation, one is maximum, another is minimum. Okay, so in this fashion, range is calculated, but it takes into account only two observations. So there arises another concept as interquartile range. So before knowing that interquartile range, you must know what is median. As I discussed earlier in the measure of central tendency, median is your middle value. Okay, median, do remember this, median is also called as 50th quartile. Median is also called as 50th quartile. Okay, because in the context of this interquartile range, it is very much important to know that fact that median is your 50th quartile. Okay, now we'll move towards the interquartile range. What is interquartile range? Interquartile range is a measure of statistical dispersion. As we all know, what exactly or how exactly the interquartile range is calculated? It is simply the difference between upper and lower quartile. Upper means 75th quartile, lower means 25th quartile. My dear friends, what I told you median is your 50th quartile, means half quartile. So 75th means your right half, and 25th means your left half. That quartile, the difference between that two is your interquartile range, which can be calculated by a formula IQR is equal to Q3 minus Q1. Okay, Q3 is your upper quartile, that is 75th quartile. Q1 is your 25th quartile. Okay, so let us take one example. I have taken here two examples, but we will take one example to solve or how the interquartile range is calculated. See, this is a data set which is given 4, 4, 10, 11, 15, 7, 14, 12 and 6. So these are one data set which is given to you. And what you have to calculate? You have to calculate the IQR. IQ and R, which is given by formula. I'm sorry, handwriting might be some fizzy. Q3 minus Q1. So always remember this formula. So what we have to do? We have to first calculate the median. And what is the first step in calculation of the median? We have to arrange that data in the ascending order. So I think first lowermost value is your 4. So I will write 4. Then again 4. Then again I think 6. Again 7. Then I think 10. What we are doing here, we are arranging the data in ascending order, which is the first prerequisite in calculation of the median. Then I think 11, after 11, 12, after 12, 14, after 12, 14, and last it is 15. Okay, so we have arranged that 15. Okay, so we have arranged the data in the ascending order. Now if we count the number of observations, how many number of observations are there? the number of observations are 1, 2, 3, 4, 5, 6, 7, 8, and 9. So if you carefully observe it, these are odd number of observation, and calculation for odd number of observation is very easy. Calculation for median when the observations are odd, it simply is the middle value. If there are 9 observations, then your middle value is your 10. Okay, 10 is your median. 10 is your median. 10 is your median, and median is nothing but your 50th quartile. 50th quartile. Okay, median is your 50th quartile. So see, median divided this data set into two halves, two equal halves. Right, 50% to the right side, 50% to the left side. So how we're going to calculate this Q3? Q3 means your 75th quartile. 75th quartile is calculated by using this. How it is calculated? You have to consider these two different half. This half which is at your right side, this half is your at the left side. So how are we going to calculate 75th quartile? Means what? We have to calculate, if we observe here 10 is your median, which has divided the data into two equal halves, 4 on the right side, 4 on the left side. So this 4 is your one part, and 4 to the right side is one part, 4 to the left side is another part. How are you going to calculate it? You just take how this 75th quartile is calculated. You have to take the average of two middle values, because see, we are calculating median here also, but in the context of these 4 different values. And if you consider 4 different values, it becomes again the even number. And how to calculate even number? You have to take the average of two middle values. Okay, so the middle values are 12 and 14. So how we're going to calculate the median of for these 4, 11, 12, 14, and 15? We have to take the values 12 plus 14 divided by 2 means what? It becomes your 13. It becomes 13. Means 13 is your 75th quartile, or your Q3, or your Q3. Okay, it is 75th quartile. Now, in same way, we will going to calculate the median for this, which will become your 25th quartile. So 4 plus 6 divided by 2 means what? Your 4 plus 6, 10 divided by 2 means it becomes 5. It becomes, I'm sorry, it becomes 5. Okay, 5 is your 25th quartile. 5 is your 25th quartile. So simply you will going to put this in the formula. Q3 means your 75th quartile, which is 13 minus Q1 is your 5. 25th quartile is your 5. 13 minus 5, it becomes 8. 8 is your interquartile range. It is very important, because range only takes into account extreme observations, only minimum and maximum. But when we calculate the interquartile range, it takes bulk of the observation, means 4 plus 6 plus 12 plus 4 plus 10. All these observations are taken into account, so it is more meaningful than the range. So this is another example which you may take as a exercise. You can practice it at home, and you can give me the answer afterwards, or even in the chat box also. Last point is bar chart, which is nothing but it is the pictorial representation of a data. Means bar chart is a diagrammatic representation of data. What is the significance of that diagrammatic representation? It makes the data more appealing, more attractive, more meaningful, and that is one of the major contribution of the bar chart. How or what exactly the bar chart is? Bar chart is the pictorial representation of a data in the form of vertical or horizontal. In this case, I have given the vertical way, where the length of all the bar is proportional to the measure of that data. Means how that data or the quantity of that data, according to that the height of that rectangle is given. Okay, so that is about the bar chart. Okay, thank you very much. I hope you all have understood what I try to deliver to you. Last Lastly, I would like to thank again, especially Dakne sir, who has given me the opportunity to present, to share what I have learned through my this learning career or learning phase. I would like to thank you all. Thank you very much. And if you are having any query, you can please feel free to ask me anytime, anywhere. I have given the contact number, email ID in the first slide itself. You can contact me anytime. I will always be there. I will try my best to help you. Thank you very much, sir. Thank you, one and all.
Thank you, sir. Your presentation was indeed very good, and we understood the concept better. Now we are opening up for question answer session. So I would request Sheetal Banerjee ma'am to start the question answer session.
Thank you, ma'am. We will begin the question and answer session with a quote. A prudent question is one half of a wisdom. Welcome everybody, all the participants to the question answer session for the first technical session of today's FDP. First question, sir, in this session is how to calculate range for grouped data.
How to calculate?
Range for grouped data.
Okay, now the range, actually group data means what? Group data means data which is given in terms of frequency. Okay, frequency or class intervals you can say. But in that case, there is a different method, slight different method by which we can calculate the range. But you will give me the chance, then I will show you later that how to calculate range. But range is very simple measure. It's simply what it does. It only takes into account two observations. It's very simple. Therefore, I told you that this method, this range is having some drawback or some flaws, that it only takes into account only the two observations. And in that group data case can also be established or can also be presented in ungrouped data, because what we have given in group data is your x variable and corresponding frequency. What is frequency there? Frequency means the number of times that particular variable has appeared. So we can also convert the data into ungrouped type, and we can also calculate the range. Thank you, ma'am.
Okay. Yes, sir. Thank you, sir. Sir, one more question is that can we include zero in mean, mode or median? Zero. Can we include zero while calculating mean, mode or median?
You can include, ma'am. You can include. You can calculate even if the zero is there in data set. You can easily calculate the mean, mode, median. No issue.
Okay. One more question is in our YouTube chat box, that if even number of observations, then how to calculate or determine the median?
Pardon, ma'am?
If there are even number of observations,
Yes.
then how to calculate?
Yes. So in case of even number, what we have to do, I told you when we calculated the interquartile range, how we are going to calculate. If say for example there are 10 different types of observations, 10 is an even number. Okay, so what you will do, first step is same, that you have to arrange the data in the ascending order. But what you will do, because you will have a question in mind, what should be the median of that even number of data set. So what we do here, we will take average of two middle values. For example, there are 10 different observations, then we will take fifth value, we will take sixth value. For fifth and sixth value, both values are added and it is divided by 2, means average of fifth and sixth value will be your median.
Yes. One more question is there, sir, that standard deviation or standard error, which one is appropriate to represent with our data set in the research article?
I think, according to my opinion, standard deviation is more appropriate because standard deviation we have performed a single set of experiment 5 times. Okay, so single set of data is there. So what standard deviation does? Standard deviation tells us about the variability of that data with the mean or a central value in that context only. But what is standard error? Standard error means it is a precision of sample mean with that of the population means there are different samples are there. I told you, I given the example that one sample from Nasik, one another from Mumbai, third from Chennai, Bangalore, and if you collected 4 different types of samples and you calculated the mean of that 4 different data sets, you will again plot that mean and you will find the mean of that mean. That will be your population mean, and you will compare that mean with the population mean. That is the concept of standard error of mean. So most of the research articles, standard error is used, but according to me, it is not that much appropriate. You simply use the standard deviation.
Yes, thank you, sir. One question is there, how to calculate precision based on range?
How to calculate
Precision, precision.
precision?
Yes,
Means I couldn't get that question.
yes, sir.
Precision
It's
means
in
what is expected there?
how to calculate precision based on range. If zero is part of observation, then only include in mean calculation.
If 0 is part of observation, then only
Then on
include. I couldn't get exactly the question what he or she is trying to ask, but 0 you can include 0. Range is simple, very simple concept. It only tells us about. It is one kind of a measure of dispersion in which there are 2 different values, extreme values. One is largest value, another is smallest value. We simply find out the difference between two. That range is also represents us a measure of dispersion. Means what? How the data is spread, how the data is dispersed. That is about range.
yes, sir. Thank you, sir. Thanks a lot for your expertise. I hope that the participants who have put their queries are satisfied. I owe my gratitude on behalf of the whole organizing team. Thank you. Thanks a lot. Over to you,
Thank you,
Alia,
ma'am.
ma'am.
Thank you, one and all.
Yeah, thank you, ma'am. So the presentation was indeed very good, and we all gained a lot of knowledge from this. So moving on to our next session. Before that, I have some announcements to make for our participants. All the participants are hereby informed that all the updates and the links, whatever is required, is updated on our official website, that is www.theijms.com. So regarding the MCQs, I have already mentioned. I am going to tell again that we are going to have a test on November 8th, sharp 7. The link will be opened for all of them to attend the MCQs, which is of 50 marks. And 20 marks is compulsory for receiving the certification for this FTP, which is definitely going to increase your API score. Now, moving ahead, let us start with our second session. And the second session, before we start, I would like to tell a good quote here that, well, Dean William R. Inges once said, the aim of education is the knowledge not of facts, but of values. So here we are with the statistical analysis and research in academics and also in our daily life, that statistics play a vital role. We cannot just get away from it. So here we have second session on the topic student t-test deciding one-tailed or two-tailed treatment by Dr. Yogesh Ashok Gadhave, Assistant Professor, Department of Agricultural Statistics, NM College of Agricultural, Navsari Agricultural University, Navsari, Gujarat. I would like Amol Jadavji to give the profile of our speaker to the participants. Over to you, Amol sir.
Yeah, thank you, ma'am. Good evening, everyone, and it's a great pleasure for me to introduce our speaker today, Dr. Yogesh Garade, sir. He is going to talk to us about the student t-test, deciding one-tailed or two-tailed treatment. Dr. Garade, sir, is currently working as the assistant professor in the Department of Agricultural Statistics at IJMS College of Agriculture, Navsari Agriculture University, Navsari, Gujarat. Dr. Garade, sir, received master degree in agricultural statistics from Govind Ballabh Pant University of Agricultural and Technology, Pantnagar, Uttarakhand, and PhD degree from BHU Varanasi in agricultural statistics. He was worked as agriculture expert at National Crop Forecast Center at New Delhi and scientist C at AP State Remote Sensing and Application Center, Hyderabad, Andhra Pradesh. He was awarded for best poster presentation in the National Symposium on Sustainable Management of Pests and Diseases in Augmenting Food and Nutritional Security, which is organized by NAU Navsari in 2019. Sir published 17 research papers in reputed journals. Sir has written 2 books and 2 university recommendations. He has attended more than 20 national, international conferences, seminars, and workshops. He is a life member of International Indian Statistical Association and Association of Agrometeorologists. Sir also worked as a guide for MSc student for the project. After this brief introduction, I request sir to give his valuable guidance to all participants. Thank you, sir. Now I hand over to you, sir. Thank you. You are not audible, sir, actually.
Hello?
Yes, sir. Now you are audible.
Okay. Thank you very much, Jadhav sir, for nicely introducing me. And I'm also thankful to organizing team International Journal of Microbial Science for a very wonderful 1 week faculty development program on statistical analysis in research. And it's definitely benefited to the students and the research workers. So today I'm going to talk on the topic student t-test, deciding one-tail and two-tail. So today's talk, we first understand what is the background of this t-test, what are the basics and fundamental of this t-test. Also, we understand what are the application and which formulas we are using in t-test. And at the last, we are working on some problem using the Microsoft Excel. Mostly the test is used for the small sample. And when your sample size is less than 30, then only we are using the t-test. And this t-test was invented by the statistician W.S. Gossett, who was working for the Brewer Guinness, is a company of the barley brewing of beverage company. And he invented the t-test for the handling the small samples for the quality control of the brewing. But the company is not allowed their employee to publish their work by their own names. That's why he published his finding under the pen name student, and that's why it is also called as student t-test. So before proceed to topic, initially we understand some terminologies which are going to be used in our today's topic, that is small sample and large sample. Let us consider the population, which is the aggregates of the samples which having the common characteristics, and from this population we draw some sample. If this sample is less than 30 or is equal to 30, then it is called small sample. And if it is more than 30, then it is called the large sample. Next term is null hypothesis and alternate hypothesis. It is most important thing in any research problem, because whenever we formulate any research, so we have to first formulate the hypothesis. Like this, for example, in here the effect of biofertilizer X on the plant growth. It means we have to study the effect of different biofertilizers on the growth of the plants. So we have to formulate the null hypothesis, that is application of the biofertilizer X do not increase the plant growth. That means it show the nullify results, or it show the null or nil nil your research. And against that we have to test it is nullify, or it is actually it is help in growth of the increase in growth of the plants. Another example I like to give here, let us consider there are 5 varieties and you have to test the average yield of these 5 varieties of the rice. Now I going to formulate my null hypothesis like this. There is no any difference between the average yield of the rice yield. It means 5 variety giving the equal yield. That is my null hypothesis. I am give the definite statement about my population parameter or my sample or my research. It is called null hypothesis. An alternative or against of this, it is called alternate hypothesis. Next important term, it comes on our research, that is significant and non-significant. And most of the cases, most of the researchers also don't know how to interpret our result. Is it significant or not? The significant, it means when you are going to test our hypothesis with respect to alternate hypothesis, and we have to compare with our standard tabulated value, and if our calculated value is greater than our tabulated value, then we call it significant. Otherwise we can call it non-significant result. Next term is degrees of freedom. Degrees of freedom, it is defined the number of restriction imposed on our population data or sample data that which cannot be varied. So that's why the number of restriction we can minus from the total number of the observation. It is called degrees of freedom. Let us consider the example. Suppose there are the 7 goggles in which are kept on the drawer, and somebody told me you have to choose the single goggle on a single day at once. So whenever I open the door at the first time, there are 7 goggles. So I have the choice of the choosing the goggles 7 times. So I choose any one of the goggles from the 7. But at the next day, it is a less than the 1 goggle. That is, there are only 6 choices are there. So accordingly, I go further. At the 7th day, there is no any choice. There is only 1 goggle is left in the drawer. So this one number, it is called a restricted parameter, and we have to deduct from our total number of the observation, and it is called the degrees of freedom. So let us consider for this example, the 7 goggles minus 1, that is 6 degrees of freedom. Next one is level of significance. Any experiment, we have to test against the different level of the significance. In most of the research program, in agriculture field, in industrial field, we are using the 5% level of the significance, because there are the more of the heterogeneity is present in our experimental unit or experimental material. But in case of the laboratory experiment, in case of the greenhouse experiment, we can test the we can keep the level of significance as a 1% level of significance. Okay, let us see the diagram. In the diagram, we have seen the shaded area is there and unshaded area is there under the curve. The shaded area is called the rejection region, and the unshaded area it is called the acceptance region. And the value which divide this both region, it is called the critical value. Now you see the whenever we formulate any experiment, we are no, my experiment is set and 100% it is true, but there may be chances it can be failure. So how many chances are there failure? So let us consider it is a 5% there are maybe failure, or 1% there may be flavor, and this is called the level of significance. Out of 100%, where say the 5% there is chance your experiment become failure. So accordingly, it is called the level of significance. Now, most important, how to decide one tail and two tail test. Let us consider the null hypothesis is mu is equal to mu0. That is to mean we have to compare. So based on this null hypothesis, we proceed for the alternate hypothesis. There are 3 kinds of the alternate hypothesis. One is mu is less than equal mu is less than mu0, mu is not equal to mu0, and mu is greater than mu0. So based on this alternate hypothesis, we can call it one-tailed test, test, two-tailed test, or one-tailed test. Again, you see the graphically, if your value is goes on negative in sense, yes, it is called left tail. I mean, if it is in positive sense, then it is called right tail. If it is placed in the both sides, that is not mu is not equal to mu0, then it is called a two-test test. Now come to our topic, t-test. Then what is t-test? Let us consider the example. There is a barley field. There are two barley fields are there, and I have to check the difference between the yield of this barley field. So I choose the sample from the field 1 and field 2. And whatever the samples I have collected, I draw the graph, and it is found the normal distributed graph, and the mean is located at the center point of the graph. Now, how to calculate this t-test? The t-test is calculated with help of the taking the ratio of the difference between the group means divided by the variability of the groups. The difference, it means the difference between the means of these two groups or two observation, and it is indicated mathematically by the mode or absolute deviation between the x1 bar minus x2 bar. And another part is variability of the group. It means how much the vary of our data from its mean. We have to calculate with help of this formula under root of s1 square divided by n1, that is a field 1, and plus s2 square divided by n2, that is for the field 2. Now there are three kind of the t-test. One is single sample t-test, second one is independent sample t-test, and third one dependent t-test. Single sample t-test, it is also called one sample t-test. Here we have to compare only one group. We have to compare the mean of the one group with respect to population mean. Another type is independent sample t-test. It is called two sample t-test. Here two groups having the two means, and there is no relationship between the two groups, or those are the independent to each other. Then it is called a two sample t-test or independent sample t-test. And last one is dependent t-sample test. It is also called pair t-test, where the observation, it can be taken from the same group, or both groups are the dependent to each other. Then only it is called, we are using dependent t-test. For example, husband wife data, left hand right side data, like that. Now for the two sample t-test, how to proceed for the test. Let us first of all, we have to ask the question to our data, that our data set is same or it is related. If it is our sample data set is same and both are related to each other, then we go for the pair t-test. If it is not, then we have to again ask the question, is there equal variances or unequal variances? And based on this size of the data, we go for the independent t-test. Now, equality of variance, it can be tested with help of the f-test. Now, there are some basics about the t-test. T-test is used when your sample size is less than 30 or equal to 30. But condition is that your population standard deviation should be unknown. And t-test is derived mathematically under the assumption of the normal distribution population. It can be found with the probability density function given as ft, that is function of the t is equal to c into bracket 1 plus t square divided by v whole bracket raised to minus v plus 1 divided by 2, where v is the degrees of freedom and c is a constant required to make our area under curve is equal to unity. Now there are some properties of t-test. First property, which is equivalent to our normal distribution, that is t-test follows the normal distribution, and it also have the bell shape or symmetrical with mean of the 0. The range of t-distribution is minus infinity to plus infinity, and the shape of the t-distribution changes as a change in the degrees of freedom. Variance is always greater than 1, and it can be defined only when the degrees of freedom is more than or equal to 3. It can be calculated as variance of t is equal to v divided by v minus 2. T-distribution always have the less peak than our normal distribution, and it's assumed to be plateautic shape. And the t-distribution comes to the normal distribution or is converted to z distribution only when n is tend to infinity. Those are the property we can understand with help of this diagram also. Let us consider this figure. In that there are three curves are there. The middlemost curve, that is a brown color curve, is a normal distributed curve, and the curve with yellow color and dark blue color, those are the t distribution curves at the different level of the degrees of freedom. Let us consider at the 5 degrees of level of the 5 degrees of freedom, the curve peak is low as compared to the normal distribution, but tail of the curve is upward than the your normal distribution. And as your degrees of freedom is goes increasing, accordingly your tail is goes down and the peak of the curve is goes up. But always for the t distribution, the peak of the curve is always lower than your normal distribution, and that is the property of a normal distribution. Range of this normal curve is again minus infinity to plus infinity, and the curve is flat equity. Now, how to find the values from the table? Let us see the table I have shown here of the t-test. We have to compare it is one tail or two tail, right? For the one tail, in the first row, it is given the value or level of significance for the one tail, and the second row, it is give the value for the two tail, level of significance for the two tail. Let us consider I have to check the value at the 5% level of significance for the two tail test. So I select the first column for the two tail 0.05, and the level of and next I have to see the uh, level of degrees of freedom. Let us consider for the 9 degrees of freedom, I have to check the value and it is comes 2.262. Similarly, I have to find the value at 5% level of significance for the one tail, then the value it becomes lower than the our previous value, that is 1.833. And if you see the table carefully, as your n or as your degrees of freedom is goes increasing and at it reaches to the z, it means when your n is tend to infinity is show your value, that is z value, and for the z value 5% level of significance it's showing 1.960. Now I'm giving some comparison to the z value. Let us consider the degrees of freedom is 10, 20, and 30, and at a level of the 5% level of significance, we are seeing the value of the t-test is goes decrease. But when your value of the degrees of freedom is more than 30, it is 1.96. It means t tends to z as n increases to infinity. Now next one, how to find the value for the two tail and one tail. Let us consider the degrees of freedom for the 2 degrees of freedom. Let us consider there are n observation, that is equal to 3. Suppose 3 observations are there. So degrees of freedom it become 2. So alpha is equal to 10. That is, I have to check the level of significance at a 10% level of significance. That's why the value for the 2 degrees of freedom is 1.886, and for the point and 5% level of significance is 2.920. Let us see the diagram. In this diagram, when our alpha is 10, so alpha by 2, it become divided by the 0.10 divided by the 2, that is 0.5. So the value for the 5% two tail alpha by 2, the value it is show for the 5% level of significance. So based on this, all those value we have seen from the table, then we have to make the conclusion based on our values which are choose from the table and which are calculated from the formula. If your calculated value from the formula is more than your tabulated value, then you can say your null hypothesis is reject and the test is significant. Now, for this application, there are number of applications in t-test. First, we have to use the t-test for the testing of hypothesis of the population mean. When your sample, when we have the single sample which is drawn from the population, and we have to compare the sample mean with respect to population mean, then we are using this t-test. Another application of t-test, that is test the hypothesis of the difference between the two mean. Let us consider there are two groups and we have to compare this mean of these two groups, then we are using the t-test. Another example, when two groups having the dependent to each other, then we are using pair t-test. Then that is another application of the t-test. The last important application of the t-test to test the hypothesis about the coefficient of correlation. Whenever we are work out the correlation coefficient, we have to test it is significant or not based on the t-test. And one more point I like to say here, t-test is only used when your sample size is less than 30. If it is not, then you proceed for the j-test. Okay, now how to calculate this test, or how to formula, what are the formulae which are using for the t-test. Now first one, if you have to test the significance of the mean of the random sample, that is called a one sample t-test, we have to first formulate the null hypothesis, that is mu is equal to mu zero. Under this null hypothesis, the test statistic is given by t is equal to absolute difference between x bar minus mu zero divided by s divided by under root of n. S divided by under root of n is nothing but the standard error of mean. Now S is calculated with help of the variance, that is nothing but variance of sample variance. So sample, in the case of sample variance, we have to divide it with help of the 1 upon n minus 1. But in case of the population variance, we have to divide it with 1 by n. All these t-test statistic is follows the t-distribution with help of the n minus 1 degrees of freedom. Now, how to compare this theoretical value with table value? If your table value at the n minus 1 degrees of freedom is less than calculated t value, then we have to reject null hypothesis, and the test we can call it significant. If opposite of this, it is called non-significance, or it is vice versa. Then how to calculate the confidence interval of the estimates? Let us consider for the one tail, we have to use the formula x bar is equal to plus or minus t alpha into s upon root of n, that is standard error of mean. Where t alpha, it is for the one tail. If you go for the two tail, we have to take the value from the t alpha by 2, and we can calculate two tail confidence interval estimate. Now, if you have to test the difference between the mean of the two samples, that is, if sample is independent to each other, then it is called a two sample t-test. For this, we have to first formulate the null hypothesis, that is mu1 is equal to mu2. Mu1, it is the mean of the first sample group, and mu2 is the mean of the second sample. These two mean we have to test with help of the test statistic, that is equal to x bar minus y bar divided by s. Here s is stand for the full variance under root into bracket 1 upon n1 plus 1 upon n2, where n1 is the sample size of the first group, and n2 is the sample size of the another group. And this test statistic follows the t-distribution with n1 plus n2 minus 2 degrees of freedom. Now full variance, it is calculated with help of the formula which is shown in your screen, that is n1 minus 1 into s1 square, that is the sample variance for the first group, and n2 minus 1 into s2 square. S2 square, it is sample variance for the second group, and the degrees of freedom which is used for this test, it is two sample t-test, it is n1 plus n2 minus 2. Another thing, when we test the difference between the mean of the two samples, when those samples are independent to each other, then it is called a pair t-test. And for this pair t-test, we are using the null hypothesis mu d is equal to 0, where d is indicate the difference of the, or it is a indicate for the pair t-test. Whenever you are formulating the pair t-test, we are using the null hypothesis mu d, right? So for testing those, we have to formulate the test statistic, and the formula is given by t is equal to absolute value of the t bar, d bar divided by s divided by root of n, where d bar is a average of the difference between the two groups, that is di equal to xi minus yi. Xi is the values of the first group, yi is the values of the second group. And here also we have to calculate the difference sample variance. Whatever the difference values we have, based on this difference value, we have to calculate the variance of the deviation. and we have to run this paired t-test. The last one and most important, that is, if you have to test the significance of the observed coefficient of correlation, we are using the t-test. Where let us consider for this test, we have to first formulate null hypothesis, that is rho is equal to 0. Rho indicates the population correlation coefficient is equal to 0. And for that, we have to formulate the test statistic, that is t is equal to absolute value of r divided by under root 1 minus r square into under root n minus 2. This t statistic follows the t distribution with n minus 2. Now, keep in mind, for the one sample t test, we are using t minus 1 degrees of freedom. For two sample t test, we are using n minus n1 plus n2 minus 2 degrees of freedom. For pair t test, we are using n minus 1 degrees of freedom. And for the coefficient of correlation, for testing the coefficient of correlation, we are using n minus 2 degrees of freedom. Now, we are work out some, we are going to solve some problems using the Microsoft Excel. It is a practical part of our today's topic. Let us first example, that is example of one sample detail. A weight reduction program claimed to effective in training obesity. To test this claim, 12 people were put on the program, and the number of the pounds of the weight gain or loss was recorded for each person up to 2 years. Then we have to understand what we have to conclude that the program is effective or not. So that's why we are going to use t-test. I'm sharing my Excel. Excel is visible, ma'am? Okay. Okay, so problem we have discussed here, there are 12 persons and the weight gain or loss, it is observed for the particular 12 persons. Now, for solving this question, first of all, we have to count how many number of samples is there. So I count it is 12. And for this counting, we have to use the formula count into bracket B4 into B15. Now next one. How to calculate mean with help of the Microsoft Excel. So we have to use the formula is equal to average into bracket. We have to give the range for which we have to calculate the mean. So in case of calculating mean, we have to use the formula average into bracket range from which we have to calculate the data. Next one, we have to require standard deviation. The standard deviation, you see the formula, that is stdev dot s. The dot s, it is indicate for the sample deviation, where the division part is 1 upon n minus 1. If you use standard deviation instead of s, there is another option is p. If you see here, the another option is p. The p is indicate for the population, entire population, and here the division part is n. So your calculation will be wrong. That's why you have to use standard deviation dot s, and it is indicate for the sample deviation or sample standard deviation. Another point, we have to calculate the standard error, that is nothing but standard deviation divided by under root of n, that is called, and we calculate it here. Now we have to place the level of significance, that is 0.05% level of significance. So we have taking here 0.05. Another one, degrees of freedom, it's nothing but n minus 1. For the one sample t test, we have to use n minus 1 degrees of freedom. Now we have to calculate t statistic. The formula which is indicate here, that is x bar minus mu, that is x bar means what we calculate 4.66 minus mu, that is we have taken the hypothetical mean, that is 0, and divided by here s, that is standard deviation 11 divided by square root of n, that is 12. So accordingly we have put all the values in the formula and we get the value 1.44. Now we have to calculate the p value. Most of the research paper, they are providing nowadays p value instead of the critical values. So This p-value we can calculate in our Excel using the formula T is equal to T dot DIS, that is distribution dot RT, right tail or left tail. Accordingly, what we have to use, we have to write here, and we have to provide the value for the degrees of freedom and the t statistic. And based on this t statistic and degrees of freedom, it show you the value of t. Another value, that is t tabulated value. It is comes with help of the formula t dot INV. In the old version of the Excel, TINV, that is a formula which is used in the old version of the Excel. If you are using updated version, you will get the formula that is T dot INV. Okay, so value, we will get the tabulated value, and based on this, suppose this is a t statistic value, this is tabulated value. Now we have to compare these two values. We have to compare these two values. That is, calculated value is less than your tabulated value, and that's why we are does not reject our null hypothesis. So we are accepting it, and that's why the test is called significant. Okay, when you accept the null hypothesis, it is called a non-significant. If we reject null hypothesis, then it is called significant. So accordingly, for the one-tail test, we are using this manual calculation. We are doing the step by step. We are calculated first mean, then we are calculated standard deviation, then we calculate degrees of freedom, we calculate t tabulated value, and based on this, we are doing all the calculation. But for the another example I'm explaining here, how to calculate with help of the Microsoft Excel using the tool data analysis. In the data analysis, if you click on the data, you will get the option data analysis at the right side of your right side top of the corner of your Excel sheet. But sometimes when you are a new user in Excel, the data analysis option will not come. So how we can take it? So first of all, we have to click on the file or function or start button of your Excel. Then go to the option. You will get another window. In that window, you have to go add-ins. Click on add-ins. You will go then bottom of your window Excel add-in. Click on go. You will get another window in which they indicate analysis toolpack, VBA, Euro currency tool, or server add-in. Out of this, you have to click on the first tab and click on OK. Automatically, the Microsoft Excel is install this toolpack in your Excel, and when you click on the data analysis option will come out. So same example for the one T-tail, we are solved with help of the data analysis. In the data analysis, you carefully see, you see there are the three options of the t-test. The first option is paid 2 sample for the mean, t-test 2 sample assuming equal variances, and t-test 2 sample assuming unequal variances. It means it is given for the only for the 2 samples, and 2 sample we have to we can test with help of this Microsoft Excel data analysis tool. But for the single sample, what we have to do? First, we have to, for this is our one sample. Okay. Now we have to create dummy sample, another sample, and it is 0, 0, right? This is a dummy sample. This is your original data for the one sample, and this is your dummy sample. Whole this calculation I did the manually as previously we have done. Same, I will count the total number of observation, mean of our data, standard deviation, standard error, t statistic, t tabulated, and all. Now I am doing same procedure with help of the data analysis tool. I am choosing the t-test 2 sample variance assuming equal variances. Okay. The first value, that is my IQ. Another is dummy variable, right? Dummy variable we have to take here as a temporary only. At the time of the result writing, we have to skip this. Now you will get the results. Oh, 1 minute, 1 second. I have to go for the unequal variances. Now see, the result for the one sample t-test, I'm creating one dummy variable and run the model or run the tab to sample t-test. But for this, while writing the result, we have to delete this column, and now see our results are same. That is t-test, the values are same, your p-value is same, and your tabulated value is also same for the one tail. Now we have to compare with help of the our calculated value and our tabulated value. If your tabulated value is greater than your calculated value is greater than your tabulated value, then test is significant. That's why I indicate here yes. Another we can see, if you see the value of p, the both values are the less than 0.05. For the 5% level of significance, whenever you are selecting the any test, it is significant or not, we have to see those values. If those values are less than 0.05, then only it is significant. Otherwise, your test is non-significant. We move further for the second problem, that is two sample t-test. Let us consider in below the data of the gain in weights of the peak fed on the two diets A and B. Now we have to test if two diets differ significantly as regard their effect on increase in weight. It means we have to check the diet effect on the group, their increase is there increasing or decreasing their weight. So this is two sample. In my presentation, I have discussed, I have to check first the variance is equal or not, and then we have to move for the t-test. If variances are equal, then we have to choose the two sample t-test using the unequal variances. If variances are not equal, then we have to use the equal variances. So for that, we have to use f-test. I'm taking the same data set and calculating the first. In the Excel itself, there is a test, f-test for the two sample of the variances. So based on this, we have to check first. So I'm taking the first data, then the variable 2, I'm taken the labels, that's why I mark here. I want to output here only, so I select it and OK. Now see, this is first of all see the formula. The formula is S1 square divided by S2 square. The larger of the two variances is placed in the numerator. It means whenever you are largest sample, you have to put in numerator. Okay, so that's why in the first diet it is showing the variance 34, and the second diet it is showing the variance 100. It means we have to again do the calculation, data analysis, f-test. Now the second part or second group is showing the highest variance. That's why we have to take first as a first variable, because we require the at place of the numerator. Okay, so accordingly I doing this. This time I want here results. Okay, now compare both. Your variance is equal, but your f value is goes change, and based on these two values, we have to decide the variances are equal or not. Now see, this is your calculated value and this is your tabulated value. If your calculated value is greater than your tabulated value, then we are rejecting the null hypothesis. Okay. Now you see your calculated value is less than your tabulated, calculated value is greater than your tabulated value. That's why the test is significant. It means there is unequal variances. So for solving this problem, again we go for the t-test, go for the t-test. Here the variances are not equal. So that's why t-test to sample assuming unequal variances. So based on f-test, we have to decide it is significant or not, or variances are equal or not. Once you decided, then go for the t-statistic assuming your sample variances is unequal. Again, I am taking here difference is 0. I will choose the label, that's why I mark here. Now I want my result here only, and then okay. Now see, the result is comes in front of you. Here is the diet A showing the mean 28 and the diet B showing the mean 30, and the variance is 34 and 100. The number of observation for the diet A is a 12, number of observation for the diet B is 22. Now, the degrees of freedom, that is n1 plus n2 minus 2, because it is a two sample t-test, and that is 23. Now I have to compare with help of those value, the test is significant or not. So based on this, you see your calculated value, it is low than your tabulated value, and that's why your result is non-significant. Okay. Similarly, you can choose this p-value. If your p-value is less than 0.05, if your p-value is less than 0.05, then only you can say it is a significant. But in that case, or in this problem, your p-value is more than 0.05, and that's why whole test is your non-significant. Now we move for the another example of the two tests where the sample size is same. Okay. Now for this also, again we have to go for the data analysis. We have to go for the, let us consider here we are taking the assuming the variance is equal. So I am taking the okay. I have to take first the first input, then another second variable, hypothesize difference is 0. I have taken here labels, that's why I click on this. Now I want the output here only, so I click on the sheet and make the okay. Okay, sorry, we are wrongly selected. Second range is okay. Now output is okay. Now see, for these two sample t-tests, assuming equal variances, you see the mean of the first variable is 50.875 and mean of the second variable is 52.875. The total number of observation are 8 and 8. So degrees of freedom is 14. Now I have to check these two values. These critical values I have to check if it is two tail or t tail. Based on this, we have to understand. See, your calculated value is greater than your tabulated value, and that's why your test is significant. Another thing, you can check your significance based on the p-value. If your p-value, I am highlighted here, that is in blue color. If your p-value is less than 0.05, then we can call it significant. Now see, though both values are less than 5, so for one tail or two tail, our test is significant. Now go for the next problem, that is paired t-test. Let us consider the school boys were given the test in statistics, and they were given the month tuition, and second test was held at the end of it. And do the mark give evidence that the students have the benefited by the ECRA coaching or not? So for this, we have to use t-test. Whenever our observation are taken, whenever our observation taken from the same group, or when our observation are dependent to each other, then only we are using the paired t-test. For t-test, again we go for the data analysis toolpath. In that we have to choose paired t-test, then OK. Select the data set for the first variable, then after the second variable, hypothetical difference is 0. I'm taken the label. Then output I want here, and I click on OK. So we get the results. So before and after, we get the mean of the both the group. We get the variance of the both group. The total number of observation are 11. This is most important. Whenever we are using the paired t-test, the sample size for the both sample, it should be equal. Then we have to get the degrees of freedom, and the another is t-test. This is a t-statistic. Now we have to compare with help of the t-tabulated. That is a critical value, and you are seeing your calculated value is greater than tabulated value, and that's why it is significant. Another, based on the p-value, we can say it is significant or not. Okay. Now those values are again less than 0.05. That's why your test is significant. So this paired test is significant. Now, last one that is most important. How to test the coefficient of regression? How to test the significance of the coefficient of correlation? So how to calculate the correlation? I'm taking the same data set before and after. Here I have to calculate the correlation. So correlation, we are using this formula is equal to correl under bracket. We have to give the range of the first group, comma, range of the another group. So we'll get the value of those. Another, you can directly calculate the correlation between the two groups using the formula, using the data analysis tool. There is correlation. Click on correlation. Select the input range. It means we have to simply select hold the data set. Here I am selected the label, so that's why I'm click on the label. Output I want here only. So okay. Now see, the both values are the equal. So you can calculate with help of the formula correl, or you can directly use the data tool, data analysis tool. Now this is a formula for the testing the significance or t-test. Now see here, R value we have calculated here. Then we have to calculate 1 minus R square. So that's why I'm using the formula SQRT, that is square root of 1 minus R square. Again, we have to use n minus 2. So that's why n minus 2 is under root n minus 2, that is 11 minus 2. And t calculated, that is absolute value of the R divided by the value of this 1 minus R under root 1 minus R square into n minus 2 under root, and you get the value 2.48. That is your calculated t value. Now tabulative value you can calculate with help of the TINV formula, and you will get the calculated table value. So based on this value, if your calculated value is greater than your tabulated value, then it is significant. So in that case, our test is significance or coefficient of correlation is affect the show the significant relationship between before or after. This is all about the doing the calculation with help of the Microsoft Excel. Apart from that, I like to share one thing here. Excuse me, ma'am, is there visible? My Chrome is visible? Yeah, I have to again share it.
No, sir, it's not visible. Chrome is not visible. Yes,
okay.
Your Excel screen is visible.
Okay, I'm sharing now. Now is it visible?
Yes, now visible.
Okay, thank you, ma'am. This is apart from this t-test, I'm telling our listeners there are number of students are there, number of research workers also there. One of the online web-based software is there, and it is definitely useful to our researcher to doing the statistical analysis and get immediate result. without proceeding to any high version software like SPSS or Microsoft Excel. We have to understand or remembering the different formulas or number of tools. So I'm click in the Google search OPStat. The software or web-based software is developed by the Hisar Agriculture University. We have to just go on the welcome to OPStat. It is a very user-friendly web-based software, and you get a number of your research data analysis with of this, and it's getting the totally free of cost, and you get immediate result for the all type of your experiment. Okay, I'm trying. Okay, anyway, ma'am, I think it is not opening because it's I think it is only open in during the office hours only, because it's a university web-based application. So I requested all the listeners, once you visit on this, on the simply type in the Google OPStat. So we'll get the link, welcome to OPStat. And there are number of tabs is given like this. I'm showing here some of the results. Like this screen will be come in your, this is a link I'm showing directly, how to calculate all those. Once you click on this website, you will get that, like this OPStat homepage. You can generate the frequency, cross tabulation is there, correlation analysis is there, comparing mean is there, one factor analysis, two factor analysis, even the pool analysis also there. Most of the researchers are interested to do the analysis in different years. Like as in our agriculture experiment, we are conducting the trials throughout the 3 or 4 years continuously, and we have do the analysis, pool analysis. In this pool analysis, this is software give the direct result. If your data set is heterogeneous or homogeneous, based on this, it can be calculated very easily. Even none of the software is given the result like this, because it's a totally manual procedure while doing the pool analysis. Because first of all, we have to understand it is homogeneous or heterogeneous of the group, and then only we have to proceed accordingly for the pool analysis. Another one important thing I like to share here, some of the researchers think if there are the more than 3 years of the data or 2 years of the data, simply we have to take the average of those values and simply run our again design, suppose RBD or CRD, but it is totally wrong. Pool analysis is totally different concept. It is totally based on our total of the sum of our treatment, and in the pool analysis we are taking the addition of the error, and based on this we have to do the our testing of our all the tests. So it is the pool analysis is nothing, it is not a average of the our treatments, it is the total of our treatment. Based on we have to proceed for the analysis of variance, and this is important thing I like to share with you. And from my side, this is uh, things I like to share with you, and I like to, one second. Thank you very much. My slide, last slide there. Thank you very much.
Indeed, sir, your presentation gave us a lot of insights into how to do stats and everything, student t-test. So it was really very good session. Next, we are going for question answer session. So question answer session is taken by Dr. Sheetal Banerjee. Over to you, ma'am.
Thank you, ma'am. Thanks a lot. It was a mesmerizing power pack session. Thank you, sir. Thanks a lot. I welcome all the participants in this question answer session of second technical session of today's faculty development program with the coach. that the important thing is not to stop questioning, as curiosity has its own reason for existing. I am putting certain questions which has been kept in the chat box by the participants for your expertise, sir. Please kindly do the needful. One of the question is that why significance level is measured at 5% level only? Is there any specific reason or is the standard for all the experiments?
No, no. First of all, it is not a standard. It is based on our what kind of experiment is going you are going to be formulate. And most of the in our case, from our agriculture background, when we are go for the field, there are different variabilities there. Climatic factor is there. Even soil fertility also vary plot to plot. At that case, in the standard, we are maintain that 5% we can take the there may be experiment maybe failure, so that's why we are taking the 5%. But most of the clinical trials is there or laboratory trials is there. We are keeping the 1%. It means there are less chances of your experiment is goes to be failure. And that's why according to what kind of experiment you are going to we conduct, sometimes there are lot of variability. Such kind we can also go for the 10% level of significance also. Point one level of the significance we also consider while doing the experiment. And in most of the in research paper also you have seen the values is indicated by the single star, 2 star or 3 star. The 3 star is nothing but the 10% level of significance.
Okay, okay, thank you, sir. One question is that can we go for t-test to know the significance of regression coefficient between two groups?
Yes, yes. See, for the regression coefficient, when you are taking the single variable, for the simple regression analysis, we go for the t-test. But for the more than multivariate or multiple regression analysis, we have to go for the f-test. And then we have come under the analysis of variance.
Okay, okay. One question is that which is the effective test of research in social sciences?
See, in social sciences is a very broad term. Okay, when you're if you are go for the survey related question and answer, or you are taking the surveys of the number of samples. So based on this, we have to first of all decide which sampling procedure we are going to be apply on social science or social science kind of the research. And based on this sampling, then we have to come to the what kind of the descriptive statistic we have to use, what kind of the result we required. Let us consider if you have the comparative studies there. The first group, suppose first farmers group, small farmers group is there. We are providing some technical knowledge to them. After that, we have to take their how they upgrade from this knowledge. Such kind of experiment we can formulate with help of the t-test.
Okay. One of the question is that can you explain pool analysis?
Yes, ma'am. So listen, most of the experiment, I'm giving the from my side or my background agriculture point of view. Let us consider there are I'm conducting the experiment in RBD, simple RBD. There are 5 treatment or a number of 3 replications are there. I conducted the trial in 1 year. Same experiment I conducted for the second or third and likewise. Now I have to compare is there year effect is there or not. Okay. But the effect of the year, it is induced in our treatment, it is yes or not. That's why we go for the pool analysis. In that pool analysis, we have to take the sum of the treatments throughout the years, and we go for the analysis of variance. But in this pool analysis, we have to keep the error term as the sum of the each year error. We have to sum at the doing the analysis of variance at the pool analysis. And this is a difference between the simple RBD and the pool analysis. And it is most of the case when you are going the long-term experiment, that case we are using this pool analysis. Another thing, if you not want to go for the pool analysis, then split plot design is also one of the option. But in that case, we have to use this year as a subplot. Then we can use for do this calculation.
Okay. Thank you, sir. Thanks a lot for your
Thank you,
expertise.
ma'am.
Thanks a lot. Means I think that your expertise, your dedication, your devotion, your hard work is a testimony. You're such a brilliant for your expertise is that. Like thanks a lot, and on behalf of the organizing team as well, I owe my gratitude to you for your esteemed presence and for your expertise. Thank
Thank
you.
you.
Thanks a
Thank you
lot.
very much. Thank you.
Very well indeed said by Sheetal Banerjee ma'am that we have gained lot of knowledge on this student test, two-tail test and everything, and hoping to do more better and stay connected with you, sir. So next, proceeding towards our end of the session, I think we have made a very good start today, and we have ended it very well also because we learned lot of things today, and the presenters were extremely wonderful to share their knowledge with all of us. And now coming to our next kind of thing announcements is that participants hereby informed that whatever queries, questions are related to your sessions, you can post it in the YouTube chat box for the answer question answer sessions. And next thing is for the updates, please do visit www.theijms.com. And next is today we are having International Stress Day. It is going to be from today to 6th of November. So stress free is a kind of thing which everybody is wanting in their life. And due to this COVID-19, everybody have been stressed out. So try to make yourself comfortable by doing meditation or yoga, and definitely it will help you in attending your sessions in a better way. And I know participants with all their daily course and all the kind of activities, they are joining to our FTP with the zeal of learning, and I appreciate to that, and I really welcome all of them for tomorrow's session as well. So tomorrow's session we have day 5, and the session 1 has replicates independence of replicate measurements, finding significant p-value, procedures for example inter-observer reliability, triangulation, and audit trails by Dr. M.R. Patil, Department of Statistics, Mahatma Phule Krishi Vidyapeeth, Rahuri. And the second session we have placate Berman design and calculations involved in it, use of Minitab software to design and interpret the results and graph by Dr. Parimita Jadhav, Department of Microbiology, Shardabai Pawar Mahila College, Varamati. So these are the two sessions with which we will be proceeding to even third session, and this is the most important aspect, I think, conduct of research projects and the research projects. So we have with us Dr. Hitesh Mehta, Sankalchand Patel University, Visnagar, for our third session on the topic conduct of research projects. So, hoping to see you all tomorrow with fresh minds to learn more new things in statistics. And definitely, we will have a special guest speaker as well, also to motivate and drive us towards learning of knowledge. Hopefully, you all will do well and stay safe during these pandemic times. Wishing you all the best and to stay stress free. Winding up and signing off Alianas from here on behalf of IJMS. Thank you, all of you.